Bio 102, Lecture 5, Osmoregulation. Good morning, students. Today we're going to look at the mysteries of osmoregulation, which is an awesome topic. We have a balancing act that we're constantly doing. The physiological systems of animals operate in a fluid environment, as you all know, and the relative concentrations of water and solutes must be maintained within fairly narrow limits to maintain homeostasis. Osmoregulation controls solute concentrations and balances water gain and loss. So this is a very nice regulatory system we have, which will control this. And excretion will get rid of the body, get rid of the nitrogenous waste in the body that we don't need anymore. So these are two acts that have to be balanced constantly and every day: osmoregulation and excretion. So we have many adaptations to this. We have to reduce water loss because that's critical. Some water is always going to be lost to metabolism, but reducing as much as we could possibly reduce, that would be a good thing. Otherwise, we'd constantly have to take in water and spend a lot of energy doing just that. So being efficient in our water loss would be the ideal way to go. So organisms have developed many adaptations. Plants have a waxy covering. Bugs have a waxy covering on their exoskeletons. There are shells made out of calcium carbonate, which will reduce the water loss on shelled animals. We have dead layers of skin that reduces our water loss. And then you could even completely go the other way and go to nocturnality, which means you're only active at night, and that way you also reduce a lot of water loss. Water is routinely lost across the skin, in urine, by feces, and from surfaces where gas is exchanged. That's just routine. But this is managed by bathing all internal cells in interstitial fluid. In insects, it's called hemolymph, or in other animals, vertebrates, it's called lymph. Metabolic byproducts must be dissolved in water. What is waste is nitrogenous products of protein and nucleic acid breakdowns. So anything that we eat will obviously have proteins or somebody else's DNA, and we've got to break it down. Enzymes break apart proteins and remove nitrogen as ammonia. Ammonia dissolves in water and becomes ammonium, which is an ion, which is very toxic and stops ATP production right away. So this is it's one way to go, but for that you would actually need to get rid of it rapidly. And the only way to get rid of it rapidly is if you live in water and you can quickly get rid of it. The type and quantity of an animal's waste products may greatly affect its water balance. Among the most significant wastes are nitrogenous products, breakdown products of proteins and nucleic acids. So some animals convert toxic ammonia to less toxic compounds prior to excretion. So ammonium, as I said, is very toxic. Endotherms produce more nitrogenous waste than ectotherms because they eat more food and they use more energy. So because they eat more food and they use more energy, they have to eat more food because they use more energy. And if they use more energy, they're going to have a lot more nitrogenous waste. So it's one of those loops. Predators in endotherms will produce more nitrogenous waste than herbivores, and that's for obvious reasons too. So osmoregulation is based largely on balancing the uptake and the loss of water and solutes. The driving force for movement of solutes in water is a concentration gradient of one or more solutes across the plasma membrane. Water enters and leaves the cell by osmosis. Now, if you remember, osmosis is a special case of diffusion where water is moving from an area of high concentration to an area of low concentration through a semi-permeable membrane. Osmolarity, that is the solute concentration of a solution, and that is what determines the movement of water across a selectively permeable membrane. And here it is again. Osmosis, the definition is the spontaneous net movement of molecules through a semi-permeable membrane to a region of higher concentration to equalize concentrations. So osmosis is a special case of diffusion, and if you go to this particular link, there's a discussion on diffusion and transport, and please do look at it and then come back to me. Osmoregulation's definition is the active regulation of osmotic pressure to maintain concentration balances, and it is a control mechanism. Here is an illustration showing you osmosis. On the left-hand side, you see a beaker that is separated by some semi-permeable membrane. On one side of the membrane is a concentrated sugar solution, so there's less water. On the other side, there is a very dilute sugar solution, and the sugar is represented by the blue dots. So after a while, if you leave this alone, what is going to happen is that the water from the right-hand side is going to start moving into the left-hand side. Sugar will not move. It's water that's going to move, and that is osmosis. So water is moving from an area of high concentration, which is the dilute solution, to an area of low concentration, which is a lot of sugar. Osmoregulation is needed because cells are more concentrated in salts than their environment. So without any regulation, water would just move on in to dilute it. And cells need to maintain their concentrations for normal activities. Cytolysis or osmotic lysis occurs when a cell bursts after excess water has moved into the cell due to an osmotic imbalance. And we want to avoid that at all costs. So this is an example of that. In the center of the picture, you see red blood cells, and they're all perfectly shaped like little donuts, and they're in an isotonic solution. So water is leaving those red blood cells, but the same amount of water is entering. So it's a balance. On the left-hand side, what you see are the same cells suspended in a hypertonic solution, so a very horribly salty solution. Well, the cells have water in them, and water is going to move from an area of high concentration, which is inside the cell, to an area of low concentration, which is outside the cell, in the futile attempt to try and balance the water gradient or get to equilibrium. It's not going to happen, so all the water will leave the cells, and the cells will shrivel up. In the right-hand side, what you see is the opposite situation where the red blood cells are now put in hypotonic solution, like maybe just distilled water. And here, now there's too much water outside the red blood cells, not enough inside. So water is going to go and try to make both sides of the membrane equal. And in this situation, the outside water is going to enter the red blood cells, and the red blood cells will keep on taking up water and keep on taking up water until the membrane will finally just pop. Just like if you overfill a balloon, it will pop. So that is in a hypotonic solution, our red blood cells will simply lyse. But the tensile strength of the membranes actually prevents that. Cytolysis occurs in a hypotonic environment when water moves into the cell by osmosis and causes the cell volume to increase to the point where it exceeds the membrane's tensile strength, and the cell bursts. The tensile strength is the maximum strength that a material can withstand while being stretched or pulled before rupturing. The compressibility of the membrane is determined by lipid compressibility, as lipids are the major constituents of the bilayer. Our own tensile strength of our cell membranes, that's between 30 and 100 atmospheres. And you might think this is overkill because we just live under 1 atmospheric pressure, and so we don't really need to be immune to 30 atmospheres or 100 atmospheres. But this is built in for extra situations which are especially stressful. Please do look at these 2 links and then come back to me. They're both very, very useful and enjoyable. Let's go to definitions. Osmolarity. So osmolarity is really difference in osmotic pressure. It is expressed as a total solute concentration in molarity, and molarity is moles of solute divided by liters of solution. So molarity, which is always abbreviated as a capital M, is equal to moles of solute divided by liters of solution. 1 mole, remember, is Avogadro's number, and that has been measured to be 6.022 times 10 to the 23. And Avogadro's number, remember, is just a unit. If you say a dozen, you automatically understand that means 12. Well, if you say Avogadro's number, you automatically know that it means 6.022 times 10 to the 23. It's just a unit of measuring how many things. Atomic mass is, however, calculated from Avogadro's number. Carbon has an atomic mass of 12, oxygen has an atomic mass of 16, and so on and so forth. And remember the periodic table, so here is the atomic mass written up. And you start out with hydrogen with 1 atomic mass, and then you keep on going up the periodic table. Here are 2 links if you need to review what Avogadro's number is and how to calculate molarity. So please do check on them and then come back to me. The unit that we are going to be using is milliosmoles per liter, because in the body we don't use osmoles per liter. That's just way too big of a number. We use milliosmoles. Seawater has an osmolarity of 1,000 milliosmoles per liter. Human blood has an osmolarity of 300 milliosmoles per liter. And the difference between hypertonic, hypotonic, hyperosmotic, and so on. Well, if concentrations on both sides of the membrane are equal, the solutions are isotonic. Some books will also say they're isoosmotic. In isotonic solutions, water molecules will cross the membrane at equal rates in both directions. If one side is more concentrated than the other, then that solution is called hypertonic, or in some books it's written as hyperosmotic. And then the net flow of water is from the hypoosmotic, which is less concentrated, to the hyperosmotic or more concentrated solution. The other solution, which is more dilute, is called hypotonic, or in some books, hypoosmotic. So here is a picture showing you that. On the left-hand side, that is the hyperosmotic side. You see more solute molecules in yellow and less water. On the right-hand side, you see less solute molecules and a lot of water, so it's hypoosmotic. And hypoosmotic means there's low solute concentration and higher free water molecule concentration. This is important because there are animals that are would like to live, well, they're aquatic animals, and they have a choice. Should we change our osmolarity and spend a lot of money on, or energy on it to do that, or should we try and keep the same osmolarity as our surroundings, just change out the solutes, the ions, and that wouldn't cost so much in terms of energy. So an osmoconformer stays in osmotic balance with its environment, like marine animals do. However, although the osmolarity is the same as seawater, the specific solutes are different and need to be actively pumped in or out. For example, concentration of magnesium ions in seawater is 50 millimolar, but a lobster has less than 9 millimolar in its circulatory fluid, so it's constantly pumping out magnesium ions, even though it has the same osmolarity in its blood as the seawater. So it's an osmoconformer. It just has different ions than seawater. Osmoconformers are isoosmotic with their surroundings, and they do not regulate their osmolarity. An osmoregulator, however, controls its internal osmolarity independent of its environment. It doesn't matter if it is or it isn't, it's just going to control it regardless. Osmoregulators can live in hypoosmotic environments by discharging water, or in hyperosmotic environments by taking in water. However, there is a big cost to this. There's a big energy cost to pump solids across the membrane. So a minimum of about 5% of our resting metabolic rate is just spent on osmoregulation. So we do it, we spend energy on it regardless of what we're doing. Marine animals, most invertebrates are osmoconformers. Many marine vertebrates and some marine invertebrates are osmoregulators. Marine bony fishes are hypoosmotic to seawater. They balance water loss by drinking large amounts of seawater and eliminating ingested salts through their gills and their kidneys. And here is a picture of a saltwater fish, and the blue depicts drinking the direction of water movement, and so the arrows going in the fish's mouth are, notice, the same size over here. Ions and water, same amount is going in the fish. Well, it will lose some water through its skin. It will actively transport ions through its gills, and then it will excrete a very salty urine with very little water. So very little urine, but whatever it is, it will be very concentrated and salty. So although it's losing some ions through its gills, it needs to lose even more through its excretion. Freshwater animals constantly take in water by osmosis from their hypoosmotic environment. They lose salts by diffusion and maintain water balance by drinking almost no water and excreting large amounts of dilute urine. Salts lost by diffusion are replaced in foods and by uptake across the gills. So this is the opposite of marine. And here is a freshwater fish. So the direction of water movement and the direction of ions, you can see it's different over here. So it'll just, the freshwater fish is actually going to take up a lot of ions in its gill and in its gills, and it will try to absorb a lot of water through its skin, and then it's going to excrete a lot of dilute urine and very little ions. So freshwater fishes will do this to adapt to their environment. Here is a comparison of freshwater and saltwater fish osmoregulation. So on the left-hand side, you see the freshwater fish. There is diffusion through the skin. So water is moving in the freshwater fish, and ions are moving out. And then it is actively pumping ions in through its gills, and it is excreting a very dilute urine. In the marine fish, what you see is water is, yes, it is diffusing through the skin because it would. The urine is awfully concentrated, and then there is active secretion, not uptake, but secretion of ions out because the fish does not want that many ions inside it. Here is a different picture. It says the same thing. It's osmoregulation in freshwater and saltwater fishes. If you prefer something that looks more realistic than a cartoon, then this will show you exactly that. There are two other terms that we need to go over just a little bit. One is called Stenohaline and euryhaline. Steno means tiny. So it cannot tolerate big changes in external environment osmolarity. So Stenohaline would be creatures that just don't like it if they're moved from, let's say, saltwater to freshwater or freshwater to saltwater. Euryhaline creatures, on the other hand, can tolerate big changes in external environment osmolarity, like salmon. They actually go from seawater to freshwater and freshwater to seawater, so they can do this. Also, I brought in this picture of a tardigrade, which you can see it undergoes desiccation, so it undergoes extreme changes in environment osmolarity, and it actually undergoes cryptobiosis, which is or practically it's anhydrobiosis, and you can actually see that the animal will live in suspended animation. You can go to the link and you can look at the tardigrade and see how it changes, and then come back to me. The energetics of osmoregulation. Osmoregulators must expend energy to maintain osmotic gradients. The amount of energy differs based on how different the animal's osmolarity is from its surroundings, how easily water and solutes move across the animal's surface, and the work required to pump solutes across the membrane. So animals will regulate the solute content of body fluid that bathes their cell. And they have transport epithelia. These are epithelial cells that are specialized for moving solids in specific directions. These cells are typically arranged into complex tubular networks. An example is in the nasal glands of marine birds, which remove excess sodium chloride from the blood. And here you see this bird, and from his beak you will actually see drops of very, very salty stuff falling out, and that is because he is trying to get rid of excess salt that he's taking in along with his food. The forms of nitrogenous waste. Well, animals excrete nitrogenous waste in different forms. They can either choose to excrete it as ammonia, so that's number one. But that would only be for freshwater animals or any animal that has access to a lot of water. They can choose to remove it as urea, and that would be somebody like us, or they can choose to remove it as a semi-solid paste called uric acid. The difference between these three is how toxic are they, and what is the energy cost in producing them. Ammonia can only be excreted by animals with lots of access to water, and those would be mostly aquatic animals. Since ammonia is very, very soluble in water, it will easily pass through membranes into the surrounding water by diffusion, so it's very energy efficient. Some animals lose ammonia over their entire body surface, like earthworms. So if you've ever picked up an earthworm from the soil and you smell it, it does smell a little stinky. Others, like fishes, will lose ammonia through their gills. Animals that excrete nitrogenous waste as ammonia will need a lot of access to water. They will release ammonia across the whole body surface. Ammonia is highly toxic and needs to be removed immediately. So the only animals who can use this method would be single-celled animals, or animals that live in water and have a very thin membrane which is in contact with water. So ammonia is very toxic, so it can only be excreted in a very, very dilute solution. Land animals don't have that much access to that much water. So mammals, some amphibians, and some sharks and turtles will actually excrete ammonia by combining it with carbon dioxide, which is also a waste product, and produce urea. So urea is less toxic than ammonia. It has low toxicity, and it can be transported by the circulatory system and stored at high concentrations pretty safely. However, it's tremendously expensive to produce, to join one molecule of ammonia to one molecule of carbon dioxide. But it is produced by the liver at the cost of 4 high-energy phosphate bonds and 5 reactions. 2 of the reactions are mitochondrial, and 3 of the reactions are cytosolic. In vertebrates, urea is produced in the liver. The circulatory system carries urea to the kidneys from the liver, where it is excreted. Conversion of ammonia to urea is energetically expensive, but excretion of urea requires less water than ammonia would. So it's the better of the two evils. Some animals go a step further and they excrete uric acid. Insects, land snails, many reptiles, including birds, mainly excrete uric acid. Uric acid is relatively non-toxic, so it's not really toxic at all. So this is awfully cool. And it does not readily dissolve in water. It can be secreted as a paste with very little water loss. Uric acid, however, is even more energetically expensive to produce than urea. So it is the most energetically expensive way of all ways to get rid of nitrogenous waste. But for the animals that use this method, it is worth it to them because they hardly ever drink water. Uric acid does not dissolve in water, so it can be excreted as a semi-solid paste, so there's very little water loss. Uric acid is excreted by insects, snails, birds, and many reptiles. However, if we have uric acid crystals, we end up getting gout, so we don't ever want to get to the point to have uric acid crystals. Here is a summary slide showing you how various animals deal with getting rid of nitrogenous waste. On the left, you see fishes. They will use a simple way of just getting rid of ammonia. Most land animals, sharks and some bony fishes, will use urine or urea. They will combine the ammonia with carbon dioxide. And then reptiles and birds and insects will actually just produce uric acid, which is horribly energetically expensive to produce, but can be secreted with very little water loss. And this is its formula. It's 2 fused rings, one 6-sided and one 5-sided. Most excretory systems produce urine by refining a filtrate derived from body fluids. So the key functions of most excretory systems are these 4. One is the excretory system has to do filtration, so it will filter body fluids. Number 2 is reabsorption, and the excretory system has to reclaim valuable solutes. It's got to get them back. Number 3 is secretion. The excretory system has to add non-essential solutes and wastes to the filtrate. And finally, number 4, the excretory system has to do excretion, which is processed filtrate containing the nitrogenous waste has to be released from the body. Here is a picture showing you how this is done in a simplified way. So these are the key 4 steps. And what you have is, in yellow, is the tube that is responsible for doing all these 4 functions. And in red is actually a capillary, which is bringing blood to the tube, giving the filtrate to it, and then the necessary things coming back out. So you reabsorb everything that you want back, and then secrete things that you actually needed to be gone, and then ultimately you would release the whatever is left over as and do excretion. Selective reabsorption recovers useful molecules in digestion. Non-essential solutes are left behind and more are pumped in by active transport to produce urine. Simpler animals use diffusion. Insects have Malpighian tubules, and we have kidneys. So we're going to look at, since this is a course on biology, we're not going to just look at ours, but we're actually going to look at do a little survey of other systems in lower animals, and then we will marvel and see, well, how close is it to us? And we will see that ours are more complex and more refined. Systems that perform basic excretory functions vary widely among animal groups. They usually involve a complex network of tubules, and they're called various names: Protonephridia, Metanephridia, Malpighian tubules, nephrons. The simplest ones are called Protonephridia, and they're found in planaria. A protonephridium is a network of dead-end tubules connected to external openings. The smallest branches of the network are capped by a cellular unit called a flame bulb. These tubules excrete a dilute fluid. And here is a picture of that. So here is the planaria. It has 2 strands of Protonephridia tubules running down itself. And then you can actually look at one of these Protonephridia here, and see that they're dead-end tubules, and they're ended with something called a flame bulb cell. Metanephridia are found in earthworms. These, since earthworms are segmented, so these have to be segmented too. Each segment of an earthworm has a pair of open-ended metanephridia, so they're not dead-end, these are open-ended. Metanephridia consist of tubules that collect coelomic fluid and produce dilute urine for excretion. And here is a picture of an earthworm and its metanephridia. And what you see is the coelom and the capillary network surrounding the yellow part, which is the collecting tubule or the unit that actually takes the liquid from the coelom, and the capillary network takes back what it needs, and then stuff is sent out. There is an internal opening which is shown here. So each segment opens into the next segment, and the external opening is just over here. So this comes, the fluid goes this way, and then this way, and then out. All right. So this is getting a little bit complex, but not so much. And then there are Malpighian tubules, which are present in arthropods and insects and bugs. They do the same thing. They remove nitrogenous waste from the hemolymph, and they function in osmoregulation. Insects produce a relatively dry waste matter, mainly uric acid, which is an important adaptation to terrestrial life. Some insects that are terrestrial can also take up water from the air, so they don't ever drink water. Here you see an insect, and you see its Malpighian tubules, and you see that the tubule actually will have, so this would be the Malpighian tubule, yeah, and this is the insect itself. So the Malpighian tubules are these finger-like things that are sticking out in the center of his stomach, and so it'll just take out the stuff that we need taken out, the insect needs taken out, and then it's excreted as feces and urine combined. Kidneys, however, are the excretory organs of vertebrates, and they function both in excretion and in osmoregulation. The numerous tubules of kidneys are highly organized, and the vertebrate excretory system also includes ducts and other structures that carry urine from the tubules out of the kidney and out of the body. So here's a picture. It shows you the human excretory system in more and more detail. So here is a person, and you see the organs themselves. So those would be the kidneys, one on either side of the spinal cord, backbone. And if you actually took this kidney and cut it open, you would see that it is organized like this. You have this layer, which is called the renal cortex. I'm sorry, this is the renal medulla over here. There's this area, and then you get this area here, which is the renal pelvis, this opening, and this is where the water will drip out and collect in the renal pelvis here, and then it's just sent out. But if you actually wanted to look at this area in big detail, this is what you would see. In the renal cortex, you would see these fine heads of nephrons, and in the renal medulla you would actually see fine tubes going up and down and back and forth and back and forth. Do notice that some of the nephrons have very long tubes, like here, and some of them are short, and we'll come back to that later in the lecture. Kidneys are responsible for filtering out urea from our blood. They consist of tubules or nephrons. The sequence of filtration starts at the Bowman's capsule. Blood pressure will force fluid from blood capillaries into Bowman's capsule. Once it gets into Bowman's capsule, it becomes filtrate, or it is called filtrate. Filtrate will never have red blood cells. It will only have everything else except red blood cells. The filtrate will then pass through the proximal tubule, Henle's loop, distal tubule, to the collecting duct, and then travel to the bladder for storage and eventual expulsion. And let's look at every step of this. So again, here is the renal cortex. This would be this area, this light pink area. These are the long tubes, so this is the renal medulla. And then the empty area here is the renal pelvis, where the drops of urine will fall and they will collect and go down the ureter to the bladder. This is how one nephron is organized. So you have the Bowman's capsule. It is like a cup, and in the cup sits a ball of capillaries, which is depicted in red. The blood pressure will push the blood, the liquid out of the capillary into Bowman's capsule, and then the liquid, which is now called the filtrate, will go into the proximal tubule and then wend its way down the loop of Henle and up the ascending tubule and into the distal tubule, and then go into the collecting duct. So let's look at this in a diagrammatic way. This is a much cleaner way of looking at it. So again, this is the Bowman's capsule, proximal tubule, descending loop of Henle, ascending loop of Henle, and distal tubule, and then the collecting duct. Most filtration occurs in the glomerulus or the Bowman's capsule. Blood pressure forces water, salt, glucose, amino acids, and urea into Bowman's capsule. Proteins and blood cells are too large to cross the membrane, so they remain in the blood. The fluid that enters the renal tubule is called the filtrate. Okay. As the filtrate flows through the renal tubule, most of the water and nutrients are reabsorbed back into the blood. The concentrated fluid that remains inside the tubule is called urine. Urine is collected by collecting ducts and lands in the renal pelvis, from where it is taken to the bladder for temporary storage and eventual expulsion. So let's look at it step by step. Bowman's capsule. The filtrate produced in Bowman's capsule contains salts, glucose, amino acids, vitamins, nitrogenous waste, and other small molecules. These things, we need them, so we're going to take them back. We don't need this. Not sure what these are, but we probably don't need those. And we probably don't need all of these. But we do need these back, so let's do that. And that would be in the next step. But let's look at it. And how does this actually work? So here is the renal artery branching into a bunch of capillaries and leaving as the renal vein. And so with the blood pressure force, it's forcing all the blood except the cells and big proteins. So they will move potassium, urea, glucose, calcium, chloride, sodium chloride, water. All of this is going to move into the Bowman's capsule. And this shows you that there are two types of nephrons. Remember I said, please remember it. Some with the regular sized loops of Henle and some with really, really long sized loops. So those that live in dry environments will have very, very long loops of Henle, so they can absorb more water back. So let's start with the sequence. From Bowman's capsule to proximal tubule. In the Bowman's capsule, the osmolarity is going to be the same as the capillaries, which is 300 milliosmoles per liter. In the proximal tubule, what will happen is reabsorption of ions will happen. So things that we want back, we're going to take back. Salt is going to be actively pumped back into the capillaries. We don't want to lose a lot of it. And because of that, it will take water, glucose, amino acids, potassium, and bicarbonate back into the blood. So then what is left is going to be urea, ammonia, and maybe prescription drugs if we're taking any. In the proximal tubule, there's reabsorption of ions, water, and nutrients. Molecules are transported actively and passively from the filtrate into the interstitial fluid and then capillaries. As the filtrate passes through the proximal tubules, materials to be excreted become more and more concentrated. Some toxic materials are actively secreted into the filtrate at this point. So here is another picture of what moves in and out and where. Amino acids and glucose, we want them back. So in the proximal tubule, they come out. They come right back. We sent them out here, which was an oops, but we got a grab. And then now we have time to adjust. We're going to pump out sodium, because if we pump out sodium, it will actually help move glucose and amino acids out along with water. And then we go down the descending loop of Henle, and a lot of water is going to move out. We go up the ascending loop of Henle. No water will move up, but only sodium will move out. And then we'll go to the distal loop of Henle, the distal tubule. And here, again, we're going to start to balance. What do we need? How much do we need? Water is still going to be pulled out. And this is where control will happen, like how much do we need to, so how much do we want to excrete of what? And hormones will act here. And then the collecting duct, where the final passage of the filtrate is, and still water is moving out. So we're reclaiming water almost all the way, except in the ascending limb of Henle. And here is another picture showing you this in a slightly more diagrammatic way, so it makes a little bit more sense. So this shows you, this is the cortex part, this is the outer medulla, and that's the inner medulla. And if we look at it this way, it's much easier to break it up and to see what is happening and what is moving out and where. In the descending limb of the loop of Henle, a reabsorption of water continues through channels formed by aquaporin proteins. Movement is driven by the high osmolarity of the interstitial fluid, which is hyperosmotic to the filtrate. The filtrate becomes increasingly concentrated as it goes down the descending loop of Henle. In the descending loop of Henle, water is reabsorbed and excretory fluid gets more concentrated, but in the ascending limb of the loop of Henle, the membrane is now impermeable to water. So the only thing that is moved out is sodium chloride, first by diffusion and then by active transport, so the excretory fluid becomes more and more dilute. Aquaporins. Well, what are they? Water molecules can move through the aquaporins in a single file, because the channel that is an aquaporin is just large enough to fit one molecule of water. So it's a very narrow channel. Aquaporins selectively conduct water molecules in and out of the cell while preventing the passage of ions and other solutes. They act in concert with the process of osmosis. So here's an aquaporin. So you can see it's awfully tiny, but it will allow water to move in and out. And again, this is the phospholipid bilayer. In the ascending loop of Henle, salt, but not water, is able to diffuse from the tubule into the interstitial fluid, and that's why the filtrate will become increasingly dilute. Because we think about it, so let's think about it. We're at the loop of Henle at the bottom. So here's the descending limb, this is the ascending limb. We're getting more and more concentrated as we get down, and water is being pulled out. Yes, that makes a lot of sense, right? So you pull water out, things are going to get more and more concentrated. However, when we go around this bend, all of a sudden we can't pull water out, but now we're going to pull out all the salt. Well, if we pull out all the salt, then whatever is left over is going to become watery. So it will become dilute. It can't become concentrated, it's just going to become dilute. In the distal tubule, this is where all the nice balances go on. So this regulates the potassium and sodium chloride concentration of body fluids. The controlled movement of ions, protons, as well as bicarbonate contributes to the regulation of pH. And then in the distal tubule, amounts of potassium and sodium chloride are regulated. Bicarbonate is reabsorbed. Potassium and hydrogen ions are secreted. In the collecting duct, there is reabsorption of water, and so urine is transported ultimately to the renal pelvis. There is 1,600 liters of blood that will flow through your kidneys per day. about 180 liters is processed by the collecting ducts to become only 1.5 liters that is going to be urine. So you can see that 1,600 liters of blood is immediately, all this water, this is moving back into the body, because by the time we get through this, from the Bowman's capsule, and we go down and up, and we're over here, and we get to the collecting duct, all 1,600 practically has been taken back, and what we're left with is just 180 liters, which is in the collecting ducts, and that is an order of magnitude less. And so at that point, we just have to reduce by 2 more orders of magnitude, and then we're done for our water saving activity. Here is another picture which shows you the concentration, which will help you understand what moves in and out and how is it more dilute or more concentrated. So if we just look at the osmolarity and we just look at the Bowman's capsule, and remember this is the capillaries that are pushing the filtrate in. The Bowman's capsule and the proximal tubule will actually have the same osmolarity as blood, which is 300 milliosmoles. But the minute we start moving things out, we start moving out water and we start moving out sugar and amino acid and so on and so forth, well, this filtrate is going to get more dilute. Yes, we understand that. And as we go down the descending loop of Henle, more and water is going out. So filtrate gets more and more dilute, concentrated. As we get to the bend of the loop of Henle, the filtrate is at its most concentrated, 1,200 milliosmoles per liter. All right, now suddenly, the ascending loop of Henle is now not able to let any water out. But we're still going to take out sodium and chloride and things like that. So suddenly, as we're pulling all the salt out, whatever is left here is going to become less dilute, more and more dilute and less concentrated. And so as we keep on going up, we're going to allow things to start moving back. That is in the distal tubule. So water will be allowed to move out, and salts will be allowed to move in or out. And we will get back to our same osmolarity as 300 as we started out with. This will happen in the distal tubule. But once we get into the collecting duct, we need to hurry up and take as much water back as we can. We've taken up most of the salts. We need to get rid of a lot of water, so we're going to do that rapidly, and the concentration or the osmolarity of the urine in the collecting duct will become 1,200 milliosmoles per liter, just like it was over here at the bend and the loop of Henle. The collecting duct carries filtrate through the medulla to the renal pelvis. One of the most important tasks is reabsorption of solutes and water. Urine is hyperosmotic to body fluids. In the collecting duct, osmosis extracts water from the filtrate as it passes from cortex to medulla and encounters interstitial fluid of increasing osmolarity. Urine produced is isoosmotic to the interstitial fluid of the inner medulla, but hyperosmotic to blood and interstitial fluids elsewhere in the body. And here is a picture showing you that. So that on the left-hand side you see the kidney, and then you see one nephron drawn for you in divisions of cortex and medullas. And so you see what is moving out, and you see that water moves out everywhere except in the ascending loop of Henle. You should know the difference between the distal tubule and the proximal tubule, like what moves out in both places. So in the proximal tubule, everything is going to be moving right back into the interstitial fluid. We want all our nutrients back, we want all our water back, and we want all our salts back. In the distal tubule, we're more selective. The sugar is already gone. We already took it out from here. So there's no sugar being absorbed in the distal tubule. The only thing that we are going to take back is, of course, water. We're always taking back water. And every time we take sodium chloride, we're going to be pulling these protons with us. and we're also going to take out some bicarbonate so the protons and potassium ions can move into the lumen or the filtrate of the distal tubule. What goes on in the medulla? So there's active and passive transport going on. Passive transport is water. Okay, so you can see water is always being passively transported. Initially, sodium chloride is passively transported in the ascending limb of the loop of Henle. Then, as we go higher up, sodium chloride is actively transported out. So what you see is the action going on in these long, thin tubes. Sodium chloride and water. That's it. Sodium chloride is actively transported out in the ascending part of the ascending loop of Henle and also throughout the collecting duct. Here is another a summary slide of all the actions, and this is now in all colors. So we went over all of these so we can just look at them, and you should stop and pause this video at this point so you can look at every single ion and see what is moving in and what is moving out where. Here is another abbreviated version. Instead of each ion written out separately, we have them in boxes, so by area. So this is the distal tubule. This is moving out in the distal tubule. This is the proximal tubule. All this stuff is moving out in the proximal tubule, and so on and so forth. So take a look. Again, pause the video over here. Take a look and make notes. Although blood osmolarity is 300 milliosmoles per liter, the kidney excretes urine, which is 4 times more concentrated. So the osmolarity of the urine increases as it travels through the nephron, generally speaking, except for the ascending loop of Henle. The kidney has the highest metabolic rates of organs due to active transport. The long loops of Henle extending deep into the medulla maintain steep concentration gradients, resulting in very concentrated urine. So mammals that inhabit dry environments have very long loops of Henle, but those that live in freshwater have relatively short loops of Henle. So these are the 2 kinds of nephrons, and they actually have different names. The ones with short loops of Henle are called cortical nephrons. The ones with long loops of Henle are called juxtamedullary nephrons. In the proximal tubule, the filtrate volume decreases as water and salt are reabsorbed. The osmolarity, however, remains the same. As the filtrate flows from to the descending limb of the loop of Henle, solids are becoming more concentrated due to water leaving the tubule by osmosis. Sodium chloride diffusing from the ascending limb maintains a high osmolarity in the interstitial fluid of the renal medulla. The loop of Henle's main function, why do we have a loop? It is to create a concentration gradient in the medulla of the kidney, allowing production of hyperosmotic urine. By means of a countercurrent multiplier system, which utilizes electrolyte pumps, the loop of Henle creates an area of high urea concentration deep in the medulla near the collecting duct. And again, what is the countercurrent exchange system? We use this, the body uses this many times in many situations, but I'm going to go through just a situation that isn't in the body, just so you understand what the mechanism is. It's to transfer some property of a fluid from one flowing current of fluid to another across a barrier, allowing one-way flow of the property between them. So if we have, let's say in a tube, some fluid that's very hot, let's say it's really hot, we're sending it, and then it's flowing in this direction. And then we have another tube right next to it, and it has really cold fluid. Well, by the time you look at the fluids on both ends, you'll actually find that the fluid on top is not going to be as hot as it was when it comes out here, because some of the heat has actually been exchanged with the second tube. And this fluid at the bottom is not going to be as cold because there is going to be some exchange going on, and that is the countercurrent exchange system, and it is used extensively in bodies and other places. So here when we look at what's going on in the loop of Henle, the countercurrent system is being used, and so here are capillaries. The red stuff is capillaries, and this is the yellow stuff is the nephron. The nephron fluid moving this way, the capillary fluid moving this way. So opposite directions, and so it's very, very easy to take water out in a system such as this. Energy is expended to actively transport sodium chloride from the filtrate in the upper part of the ascending limb. The countercurrent multiplier system involving the loop of Henle maintains a high salt concentration in the kidney. This system allows the vasa recta to supply the kidney with nutrients without interfering with the osmolarity gradient, which is very nice. Long loops of Henle extending deep into the medulla maintain steep concentration gradients, resulting in very concentrated urine. Long loops of Henle are used when the water is in short supply. The longer the loop, the bigger the concentration gradient. The further down the loop descends into the medullary tissues, the more ions like sodium and calcium ions can pass out, so the urine can get more and more concentrated. So as we can see in this particular slide, we're going to walk through what is moving in and out, and we're going to look at water, which is passively being transported out in the descending loop of Henle. And as you can see, the osmolarity is 300 as we start out in the proximal tubule, 1200 at the bend of the loop of Henle, 300 at the distal tubule, and then again 1200 in the collecting duct. And we'll look at what happens next in the sequence. And what is happening next is the ascending loop of Henle. And notice what is passively being transferred out is sodium chloride. And because it is moved out, this solution becomes dilute and more dilute, and then it's actively pumped out and gets more and more dilute because now water is not moving out. It's the same amount of water. And then when we look at what happens next in the collecting duct, water is being moved out passively throughout. And what we see is also that urea is moved out passively, and we see that sodium chloride is moved, pumped out. So I hope that helps. Hyperosmotic urine can be produced only if considerable energy is expended to transport solutes against concentration gradients. The two primary solutes affecting osmolarity are sodium chloride and urea. Energy expenditure drives what animals do with their nitrogenous waste. Freshwater fishes, amphibians, and birds, so their energy expenditure, let's just take a look at that. Marine bony fishes are hypoosmotic compared with their environment. Their kidneys have small glomeruli. Some fishes actually lack glomeruli entirely. Filtration rates are low, and very little urine is excreted, like we saw in the early slides. Freshwater fishes, they conserve salt in their distal tubules, and they excrete large amounts of very dilute urine. Amphibians, when they're on land, they conserve water by reabsorbing water from the urinary bladder. The vampire bat, which is an unusual creature, it can alternate rapidly between producing large amounts of dilute urine and small amounts of very hyperosmotic urine. Birds have shorter loops of Henle than mammals do, but conserve water by excreting uric acid instead of urea. It's kind of interesting, yes? So let's look at homeostatic regulation. A combination of nervous and hormonal controls manages the osmoregulatory functions of the mammalian kidney, and these controls contribute to homeostasis for blood pressure and for blood volume. And this is important because we're trying to move things in and out, and there has to be some control as to how much is moving in and out at any one time in case we manage to mess it up or lose what we really want to keep. So the control of osmoregulation is done by Osmoreceptors. These are cells that are in the hypothalamus of the brain. They sense the sodium concentration of the blood. The nerve endings of these osmoreceptors are located in the posterior pituitary gland, and they secrete ADH, which is antidiuretic hormone, which means less urine production. Anti meaning less, and diuretic means producing urine. So if the sodium concentration of the blood is high, the osmoreceptors will secrete antidiuretic hormone. This reduces urine volume and lowers blood osmolarity. If the sodium concentration of the blood is low, then ADH is not secreted. So that's just a feedback loop. By the hormone ADH, which is produced by the hypothalamus, this is a negative feedback loop. The set point is 300 milliosmoles per liter. ADH makes the epithelium of the kidney's collecting ducts more permeable to water. More water gets reabsorbed, urine volume decreases, urine becomes more concentrated, so then ADH secretion is reduced, and that is why it's a negative feedback loop. ADH actually works by temporarily increasing the production of aquaporins, and these are those tiny channels. Aquaporins bind to the membrane. Each aquaporin has channels to absorb water molecules. Aquaporins are the plumbing system for cells. Every cell is primarily water, but the water doesn't just sit in the cell. It moves through it in a very organized way through aquaporins or water channels. And here we can see that happening. This is a idealized collecting duct cell, and what you see is the ADH receptor actually acting on it, and storage vesicles being generated, which have aquaporins stuck on them, which then go and act on the plasma membrane, expand it, they join up to it, and because of all these aquaporins, these extra ones, now more water can move from the nephron back into the collecting duct cell and then back into the body. So here is regulation by ADH. So we start out at a set point of 300 milliosmoles per liter. And let's say we've done a lot of exercise, so the blood osmolarity, let's say, has increased a lot. Well, immediately the osmoreceptors in the hypothalamus will sense the sodium concentration in the blood, and it will trigger the release of ADH, which will go and affect the distal tubule, and will also tell us to go drink some water. And then the distal tubule will reabsorb a lot of water, so our blood osmolarity will come back to 300. There's also a second system of control, and there is the renin-angiotensin aldosterone system, or the RAAS system. That activates when blood pressure drops due to dehydration, and this is a different system. Renin is released, which cleaves angiotensinogen to angiotensin II. Angiotensin II acts like a hormone by constricting arteries or arterioles, which decreases blood supply. So this system only occurs when there is a dramatic drop in blood pressure, which is when you might have a cut. So angiotensin II also stimulates the adrenal glands to produce aldosterone, and aldosterone, which is another hormone, will act on the distal tubules of the kidney, allowing more reabsorption of sodium and water to increase the blood pressure, and this will rehydrate the organism and bring back the osmolarity to 300 milliosmoles per liter. So this is the RAAS system, and it shows you how this works, and how we get back to our 300 milliosmoles per liter. We do need two different negative feedback loops. It's not for redundancy, but for different problems. A massive blood loss due to an open wound will activate the RAAS system, although ADH will not act. ADH will decrease urine output and increase urine osmolarity, and so does RAAS. But notice that the reasons are different. Oh, and I brought in this cartoon. Well, it's actually a picture from a very old movie called Lawrence of Arabia, which was based on a real person who went around in the desert of Egypt, and all you see are these beautiful pictures of the beautiful desert, and not a drop of water in sight. And this guy is on a camel, wandering around in the desert, and it doesn't have water, so he's saying, thank goodness we have ADH. We do have other checks and balances, because if we are trying to set the osmolarity at 300 milliosmoles, and that's great because, they're for different reasons when there's not enough water. But what if we drink too much water? Then we still need something to manage that. So there's another hormone called ANP, which opposes the RAAS system. ANP stands for atrial natriuretic peptide. Atrial, remember, is the heart. Natri stands for sodium. Uretic means urine. So it is a peptide, which is a protein. And ANP will inhibit the release of renin. It will inhibit the sodium chloride reabsorption by collecting ducts, and it reduces aldosterone production by adrenal glands. And it is a vasodilator, so it will reduce water in the circulatory system. It will reduce your blood pressure and does exactly the opposite of aldosterone. So ANP will generate sodium ion loss. It is produced by the heart. The overall effect of ANP on the body is to counter increases in blood pressure and volume caused by the RAAS system. So here is the renal response to volume concentration. There are several checks and balances. In the center you can see our nephron, and it is acted by various enzymes, hormones, ADH, ANP. Notice the arrows are different colors: aldosterone and angiotensin and sodium resorption. Here is the renal response to volume expansion. Notice that the arrows are now, look at the arrows, they're different. All right. And the last thing I wanted to talk about were adaptations. Birds do not have long loops of Henle, so urine cannot be as highly concentrated as mammals, and they produce uric acid as their main excretory product. Freshwater fishes produce dilute urine, and a lot of it. Amphibians produce dilute urine in freshwater and concentrated urine when on land. Marine fishes produce very little urine, have small or absent glomeruli, absent distal tubules, and few nephrons, which are very small. Marine fishes use mainly kidneys to get rid of sulfates, calcium ions, magnesium ions, because they keep drinking salt water. Here is a summary slide. It shows you what animal. So the first one shows you a freshwater fish, and it tells you it doesn't drink water and it tries to get the salt out. But it produces large amounts of urine, and the urine is less concentrated in the body fluids. And then you go on to the marine bony fishes. They drink in water. They want salt out, water out. They produce very small amounts of urine. And then the last one is terrestrial vertebrates. So we produce a moderate amount of urine. So I hope this was very helpful, and I will see you next time, students.